Welcome to an edition of CCSD Living Environment. Today we're going to talk about a process called meiosis. We know that in an organism with two parents, we inherit one copy of every gene from both of our parents. So we got one from mom and one from dad. So then when we go to pass on the information to our offspring, what needs to happen? Well, we need to take what we inherited from our parents and we need to separate those genes, so that when we pass information on, we're only passing one copy of the gene on to our offspring. So in the production of the gametes, the sperm or the egg, we need to make sure that we're separating what we got from our parents, so we're only passing on one copy. Well, the chromosomes that we have in our cell nucleus are carriers of genes. Remember, the genes are found on the chromosomes. The genes are located in very specific places on each chromosome, and those places are called loci or loci. It's just a location on the chromosome where you find that gene. It's the position of the gene on the chromosome. So it gives you a picture. So here's the copy of the gene that I got from my mom. Here's the copy of the gene that I got from my dad. This happens to have the P gene, the A gene, and the B gene. Now, they have the same genes, as they should, and so the locations where those genes are what we refer to as the loci. It's just where that gene is found. Now, let's review for a minute. If I inherit big P from my mom and big P from my dad, then I am homozygous dominant for the P gene. If I inherit little a from my mom and little a from my dad, I'm homozygous recessive for the A gene. And if I inherit big B from my mom and little b from my dad, then I'm heterozygous for the B gene. So just review of some of those terms. So what are we going to call these chromosomes? These chromosomes we refer to as homologous chromosomes. In other words, they're the same size, they're the same shape, and they have the same genes for the same traits. The chromosomes from the male parent has to correspond to a chromosome from the female parent. You get one of each chromosome from both parents. So whatever you get from one, you have to have a matching copy from the other parent. In this case, because we got both of them, all of our cells have both of those chromosomes. So we call those diploid cells. It's a cell that contains both of the homologous chromosomes. Diploid means two sets, di means two. So diploid is represented as 2n. And for humans, the 2n number is equal to 46. So if you're asking why 2n, well, what do you think n represents? N is a single set that's found in the sperm or the egg. So if here's my egg cell with n for a chromosome number, which in a human is 23, and my sperm cell, which has n for a chromosome number, which again in a human is 23, when I put those together in my fertilized cell, which would be called a zygote, that would give me 2n or 46. So let's look at the math that's happening. I've got n plus n, which gives me 2n. So 23 plus 23 gives me 46. So in those cells, what I have is the diploid, or the 2n number. In other words, I have the copy from my mom and the copy from my dad. Okay, so what do we call those cells that only have one copy? The gametes that we produce, the sperm and the egg. Those are referred to as haploid or monoploid cells. They're cells that contain a single set of chromosomes. The gametes of sexually reproducing organisms are haploid. Think about that for a minute. If you're going to pass on your genetic information and you're only giving half, then that means that you have to be giving a half of what your cells have. So if all of our cells have 46, then I can only pass on 23 chromosomes to my offspring. So the haploid number is represented as N. And for humans, we said N is equal to 23. So if I take my egg cell that has one chromosome now, remember that homologous pair of chromosomes got split up when I made the egg and the sperm. So I have one chromosome now here, and then my sperm has one chromosome. So I have the A gene and the A gene. When that gets passed on to the zygote, now here's my gene pair. In the zygote, I have the homologous pair of chromosomes. So this would be haploid cell, diploid cell. Or it would be the N cell, and the 2N cell. So that's kind of the background of meiosis and what needs to happen and why we need to go from that diploid number, that 2N number, a full set of chromosomes, to half in our gametes, half in the eggs and sperm. That's the reason why. Now what we need to look at are the phases of meiosis. How do we actually do it? So meiosis is the process where we're actually going to cut that chromosome number in half. Remember, I started with 46. I need to end up with 23 in the eggs and sperm. So I need to cut that chromosome number in half. So to do that, the haploid or N gamete cells are produced from a diploid or 2N cell. And that should make sense to you. We have to start with the diploid cell because that's what we have, that's what we got. So I start with that 2N cell and I'm going to split that and end up with cells that only have N for the chromosome number. Well, let's look at how we do that. So meiosis is a process where the number of chromosomes per cell is cut in half through the separation of homologous chromosomes in a diploid cell. So the purpose of meiosis is to cut the chromosome number in half. Meiosis involves two divisions, meiosis 1 and meiosis 2. At the end of meiosis 2, the diploid cell becomes 4 haploid cells. Think about mitosis for just 1 minute. We start with a 2N cell, we end up with 2 cells, both that have 2N for a chromosome number, right? Identical cells. So if splitting it once gave me 2 identical cells, what do I need to do? I need to split it a second time so I end up with 4 cells that have half the number of chromosomes. So let's take a look at the actual process. In meiosis 1, there is an interphase, and what took place during interphase? During interphase, the chromosomes replicate. Prophase 1. Well, we know that the nuclear membrane is going to disappear. That stays the same, but there's something different that happens now. Each replicated chromosome, so think about that. You started off with 1 chromosome from mom, and you have 1 chromosome from dad. During meiosis 1, those chromosomes are going to replicate. So now, remember, we have double-stranded chromosomes. Those double-stranded chromosomes are going to come together. That didn't happen during mitosis. They come together and they form something called a tetrad. Tetra means 4. How many do I have? 1, 2, 3, 4. So each replicated chromosome pairs with its homologous chromosome, and that pairing forms a tetrad. Tetra means 4. Okay? So now that they've done that, a process called crossing over can take place. Crossing over, when the tetrad forms during prophase 1, the homologous chromosomes can exchange information at their tips to produce new combinations of alleles. It leads to greater genetic variety. So here's the one from my mom that replicated, the one from my dad that replicated, and what they'll do is they'll come together enough so they can exchange their tips. And when that happens, here's some of dad's DNA on the chromosome from mom and some of mom's DNA on the chromosome from dad. So I get more genetic variety that way. What happens during metaphase? Well, during metaphase, the paired homologous chromosomes are going to line up in the middle. Sounds very familiar. During anaphase, the tetrad actually separates. So that means the tetrad is going to pull apart 2 are going to go toward one pole, 2 are going to go toward the other pole. That process is called disjunction, when they separate. And the spindle fibers pull those homologous chromosomes toward the poles. So again, I have one from mom, one from dad. They're going to replicate. They come together. Remember what we said before. When they come together, there's one centromere. Because there's only one centromere, how many chromosomes do I have? Yeah, only one chromosome at that point, but it's got 4 legs to it. That's the part that's different. So during telophase, a nuclear membrane will form around each group of chromosomes, so I have 2 cells. So here's my 2 cells. What type of chromosome is in both cells? That's right, double-stranded chromosomes. So in my picture, I start out with a 2N cell, and I get 2N cells. But what's the problem? These cells both have double-stranded chromosomes. And remember, we don't want double-stranded chromosomes. What do we want? Single-stranded chromosomes. So because I go from the 2N cell to the N cell, we call meiosis 1 the reduction division, because it's at that point where the number of chromosomes actually gets cut in half. So at the end of meiosis 1, 2 daughter cells are produced that have half the number of chromosomes as the parent cell. Meiosis 1 is known as the reduction division because the chromosome number goes from 2N to N. The cells produced have double-stranded chromosomes. Remember, we don't want double-stranded chromosomes. The same reason why in mitosis we had to undergo the rest of that process, because what type of chromosomes do we want? Single-stranded chromosomes. So the whole purpose behind meiosis 2 is to get single-stranded chromosomes. So there's no interphase. I don't want the DNA to replicate again. I don't want the chromosomes to replicate again. I go to prophase 2. The chromosomes become visible. No tetrads are formed. Homologous pairs separated in meiosis 1. Those homologous pairs that had come together pulled apart. So they're double-stranded, but there's only one chromosome in each cell. That's the difference. So in metaphase 2, the chromosomes are going to line up in the center of the cell. During anaphase 2, the paired chromosomes are going to separate. And remember, separation in this case during anaphase we call disjunction because they're coming apart. Telophase 2, the nuclear membrane reappears and both cells will divide to produce 2 daughter cells. And those 2 daughter cells have the haploid number of chromosomes. So let's look at it. If this is my original cell, so my 2N cell in this case, the chromosome number is 2. One from my mom, one from my dad. It had to replicate. They came together. They came apart, disjunction during anaphase. Telophase, I end up with 2 cells, both with double-stranded chromosomes, and look what's happened to my chromosome number. I've cut my chromosome number in half. So what needs to happen? I need to get rid of the double-stranded chromosomes, so it undergoes the process again, and now I end up with 4 cells, all single-stranded chromosomes, and the chromosome number has been cut in half. I've gone from my 2N cell, where I had 2 chromosomes, to each one of my N cells with 1 chromosome. 4 daughter cells are produced at the end of meiosis 2. The cells produced have single-stranded chromosomes, which is what I want. So when you take one of these cells, one of these gametes, which could be an egg or a sperm, and they get passed on, when that egg or sperm meets up with the other egg or sperm, and they come together, N plus N gives us back the 2N in the fertilized egg cell. So that takes you through the explanation of how we go from a 2N cell to N cells in the process of meiosis. And again, that occurs in sexually reproducing organisms. Thanks for watching, and I'll see you next time.